Welcome to Unscripted, the AMCP podcast, a look inside managed care pharmacy.
Though it sometimes doesn't feel like it, healthcare providers and health insurers are working towards the same goals to ensure that patients receive appropriate care. But what if different stakeholders in the healthcare ecosystem can't really agree on what's appropriate? If the guidelines and the best available evidence don't tell a convincing enough story, payers may have a hard time justifying coverage for therapies or biomarker testing, which is the focus for today. It all comes down to manufacturers not only demonstrating clinical utility, but also ensuring that the clinical utility that the test offers is what payers are looking for. Welcome to Unscripted, the AMCP podcast. I'm Steve Kheloussi. I'm the principal consultant with Kheloussi Consulting and a managed care pharmacist by background. In this episode, we'll be discussing clinical utility, what it is, and how it contributes to whether patients receive coverage for biomarker testing. We'll be discussing this from the perspective of precision medicine and oncology, but clinical utility is a broad term that can really apply to any intervention that payers are considering covering. Today, I have the pleasure of speaking with Dr. John Fox, Senior Medical Director for the Americas, which he tells me extends from the Arctic to the Antarctic at Illumina. Welcome, John.
Thank you, Steve.
So, John, I gave a short introduction, but why don't you tell the audience a little bit more about yourself?
Sure. First of all, I'm a pediatrician by training. I worked as a practicing pediatrician for about 5 years, but I've also worked for the Centers for Disease Control as a chronic disease epidemiologist. My son would say that was in the last millennia, Dad. For the last 20 or so years, I worked as a health plan medical director, reviewing technology, writing medical policies, and reviewing the things that we're going to talk about today regarding testing, including analytic validity, clinical validity, and clinical utility. For the last 5 years, I've worked in the market access position here at Illumina, which is the world's largest manufacturer of sequencing equipment.
Fantastic. Thank you. And I think it would be a good idea to address the elephant, or rather the fish in the room first.
Yeah.
So can you tell me about the fish behind you?
The fish is a sailfish that my son at the age of 14 caught. He's now 28 and still refuses to take it from my office to put it in his house, but it's a nice conversation piece.
I love it. It absolutely is. All right, great. Well, let's jump right in with, I think, what should be a simple question, but I know it isn't. What is clinical utility?
Yeah. Well, clinical utility is a conundrum for everyone because we don't have agreed upon definitions. But clinical utility is a function of two predecessor functions: analytic validity and clinical validity. Analytic validity is something that a laboratory establishes, and it's simply the ability of a test to accurately detect the presence or absence of a biomarker. And I use biomarker and genomic very interchangeably. And so the question is, well, how do you establish analytic validity? Well, first of all, you have to have a known sample that has that biomarker, whether that be a contrived or spiked sample, or from a tissue where we've established independently that biomarker is present. And then that lab establishes the ability of its laboratory developed test, or in some cases, an in vitro diagnostic, to detect that biomarker. And I think that the key thing here that hangs payers up sometime is that doing this type of analytic validation is really the baseline standard of care in any CLIA certified lab that follows CAP guidelines and AMP guidelines. So you can't run a lab unless you're doing that analytic validation.
Great. So you mentioned a few acronyms that I think would be valuable to define for the audience. So you mentioned CAP and AMP. Why don't we start there?
Yeah. So CAP is the College of American Pathologists, and that organization establishes what needs to be measured as part of analytic validation, including accuracy, level of detection, sensitivity, specificity. AMP, on the other hand, the Association of Molecular Pathologists, establishes how to actually do that. So CAP establishes what needs to be measured, AMP how to measure it.
All right. Well, thanks for that overview, John. And you mentioned analytical validity, and how about clinical validity?
So clinical validity first has nothing to do with a laboratory test. It's simply a measure of how the genomic variant or the biomarker correlates with the clinical condition or outcome. So it's measured by clinical trials, case control studies, cohort studies, large clinical databases. For example, if a variant or a biomarker is present in 60% of patients with the disease and 1% of controls, we can calculate an odds ratio. I've actually done that. And that odds ratio is 148. So the chances of having the disease are 148 times higher if you have that biomarker than if you don't. And so the clinical validity, therefore, is a function of the scientific evidence linking that biomarker, that genomic variance to the disease. Again, it's not a function of the laboratory test, it's a function of the clinical evidence.
Okay, great. And so these two terms then are precursors for clinical utility. Is that correct?
Yes. We want to know that we can detect the biomarker and that the biomarker we're detecting has some clinical relationship to the disease in question.
And then we can start to determine the clinical utility of the test. And so what do we mean by clinical utility in that case?
In my experience as a health plan medical director, clinical utility really answers two questions. Does the result of the test change management? And does the change in management improve net health outcomes? And so importantly, the clinical utility is not a property of the test itself, but rather a function of the genomic variant. So when the FDA reviews a test and says there is a relationship between the presence of this biomarker and the use of a drug and disease outcome, we're really establishing the utility of that biomarker and predicting response to a drug.
Interesting. And so there's a lot to dissect there. When we're looking at improving outcomes, what outcomes are we talking about?
Yeah, well, that's the $1 million question. Even the FDA doesn't have a standard definition of health outcomes, but it says that it has to improve how a patient feels, how a patient functions, or survival. But there's not a standard definition. And I think that's where the complication comes. There are different definitions of improved health outcomes that we may disagree on the clinical relevance of those. So for example, if a drug or a therapy just improves how somebody feels or delays progression of cancer, but doesn't improve survival, is that an improved net health outcome? And that's a question that we don't always agree on.
That's a really interesting point. It's not necessarily that different stakeholders can't agree on what clinical utility means exactly, but it's more the outcomes that we're talking about.
It's the relevance and the meaningfulness of that net health outcome and how we define that. I'll give you another example. This is outside of cancer, but in the pediatric rare disease space, we have whole genome sequencing for the kids who have developmental delay or autism with other features or global developmental delay. And we can establish a diagnosis in 50% of those kids. And payers will say, well, if you can't do anything about it, why does it matter? Well, there are a lot of other benefits to what we call ending the diagnostic odyssey, where kids undergo repeated tests or repeated visits to specialists to try and figure out what's going wrong. One of those is, as I said, ending all the testing that's going on. And outcomes may not result in a drug therapy. It may result in a change in management of the patient, a change in diet, for example, by supplementing the diet. It can certainly result in discontinuation of ineffective therapies. And there are certainly other benefits for the parents too. There is reproductive information that's available. They can connect with social networks on the internet with kids who have exactly their diagnosis. And it also helps the parents pivot from what's wrong with my kid to what can I do for my kid? So if we simply look at health outcomes in rare disease, we don't know what the health outcomes are because it's rare. We can't do a randomized clinical trial typically, but there are other health benefits, and those other health benefits are where there's disagreement on, are those important? Are they relevant? And for payers who cover a family, I would argue that there's clinical utility in the family knowing the diagnosis in addition to the utility for a kiddo.
So that creates a real challenge, I think, for the payer, right? Because they are concerned about a number of things, not only the well-being of the patients, but also the bottom line. And I think when you start to take that into consideration, you're no longer talking about clinical utility, you're talking about the economic feasibility of the test and whether it's worth covering, which you had alluded to previously, that the economic considerations come after, they're adjacent to the conversation about clinical utility.
Yeah.
Is that right?
Yeah. Right. And it's a bit aggravating, honestly, for me, Steve, as a pediatrician, to hold testing to a different standard than we do for treatment for other conditions. We were chatting earlier about the payment for joint replacement, which is the most costly procedure in the United States today, between hips and knees and shoulders and ankles. And there is no evidence that improves survival, but it certainly improves quality of life at a significant cost. If we say, well, we'll only pay for diagnostic testing if it results in treatments that reduce the total cost of care, then it's likely we wouldn't pay for any testing. And that's an untenable position to put kids and their families in if we say we'll only pay for testing if it reduces the total cost of care.
That's certainly a challenge. But would you say, based on the availability of biomarker tests that are out there today, maybe some that are in the pipeline coming in the future, that they do hold treatment guiding value?
Yes, I think so. And I think the other area where there's challenge, let's go back to cancer, is the definition of clinical utility in cancer. We typically associate that with, we have a drug and the response to that drug is predicted by a biomarker, and that response to the drug results in improved overall survival. That's one mechanism for assessing utility, but there are others, probably the biggest of which I think is the ability to decide whether or not we want to continue with therapy or not, but also whether or not we want to participate in a clinical trial. And most payers will say, I'm only interested in a companion diagnostic that establishes whether or not somebody's likely to benefit from a drug. So a lot of payers in the United States today will say, we'll pay for a test that has a companion diagnostic indication, but we won't pay for any other lab developed test. And this gets back to the heart of our discussion of analytic validity. That biomarker that predicts a response to the drug, the utility is inherent to the biomarker, not the test. If I can, with good analytic validation, with high specificity and sensitivity, detect that biomarker, it really doesn't matter what test it's in. Mm-hmm. Again, we've gone through in a CLIA certified lab through the CAP and AMP review process, and we've established that we can identify using any methodology, we can identify an ALK fusion, for example. So even though there's one test that was approved by the FDA, the analytic validity carries over from any test to that biomarker. We've already said that the clinical validity isn't a function of the test, it's a function of the clinical studies. And so a lot of payers will say, I only pay for a test with a CDX indication, but that's not consistent with the evidence that shows if you can show you can identify that biomarker reliably and accurately, you should be able to use any test. Probably the best correlate I can give is with imaging studies. So that years ago, we did studies to show that CT scan, we could identify smokers who had early stage lung cancer. Nobody's asking, was that done on a Siemens CT machine or a General Electric machine? And I think the same is true here. As long as we can establish there's analytic validation, it is irrelevant what test platform it was done on.
Yeah, wow, that's fascinating. So it sounds like the payers just in general are not questioning the clinical utility of FDA-approved companion diagnostic testing.
Generally, no.
It becomes more of a question with the lab-developed tests, which have, of course, shown that clinical and analytical validity. And it's now a question of what is the outcome that that test is going to potentially improve, and does that matter to the payer? Sometimes I think there's a disconnect, right, between the stakeholders, we've talked about this already, that not only is it something that could be important to the patient, to the patient's family, to the provider, but from the manufacturer perspective, this is also a challenge, because you would love for your tests to be covered by payers, and payers want to cover tests that are going to improve clinical utility, improve outcomes. But there is sometimes a disagreement even between the maker of the test, the biotechnology company, and the payer. So can you talk a little bit about that?
Probably the biggest issue in this space, honestly, Steve, is a disagreement about whether or not there is enhanced utility in a large panel, say a 300, 500, 800 gene panel versus a less than 50. And less than 50, as most people know, is associated with a code that says this test has 50 genes or less. Is there increased clinical utility for a large panel versus a small panel? There is a recent publication that showed that most less than 50 gene panels, over 94% of them, don't capture all the NCCN recommended biomarkers. And in which case, if you're running a test that doesn't capture all those, you may miss opportunities for targeted therapies. And in general, targeted therapies have been shown to improve net health outcomes, overall survival, or at least progression-free survival compared to non-targeted therapies.
So the targeted therapies are not typically going to be first line. And so patients are going to be treated with other broad therapies first. And I think there's some data that we've talked about in the past that shows that earlier testing, earlier targeted treatment can actually benefit patients in terms of less disease progression and less adverse outcomes, less adverse events from therapies that aren't making as big a difference as the targeted therapies would, right?
Yeah. Yeah. Well, ideally, when we say net health outcome, we want to maximize the benefit and minimize the harm. And when we don't do adequate testing in cancer to determine what optimal therapy is, there is the potential not only to introduce harm because patients are on a therapy that doesn't produce the net survival benefit that another therapy would, but there's also potential for waste where payers and patients are paying good dollars for a therapy that we could have predicted wouldn't work.
Yeah, so it's not only the therapy and the impacts of the patient going through that unnecessary therapy, but also the payer paying for an unnecessary therapy. And so there could be some economic utility, if you will, to earlier testing, even if it's not necessarily an outcome that they're as interested in traditionally.
Right. It's interesting. A lot of the economic analyses that have been done looking at economic utility show that, and it's mixed. Some studies show that large panel testing and treatment results in an acceptable cost effectiveness ratio and others don't. But the key is that we're tying drug and treatment together. But if you just look at the cost of a diagnostic test to establish what the right therapy is, it's very affordable. It's around $5,000. Testing is just a small part, let's say $3,000 compared to spending $30,000 to $50,000 a month, every month until the patient progresses or has been determined not to be responding.
You were previously talking about the less than 50 gene panels versus the larger panels and the considerations that payers have to think about there.
Yeah. So the first point is that a large panel is much more likely to identify all the relevant FDA-approved treatment options for a patient compared to a small panel. The second important thing is that for a patient and a physician who are interested in knowing all the relevant treatment options, including clinical trials, large panels are twice as likely or more than twice as likely to identify all the relevant clinical trials that a patient may be eligible for. And while they may not be all first-line options, in some cancer types there are, where there aren't good treatment options. But more importantly, the NCCN and others recommend that all the testing be done up front for several reasons. One, you want to know what all the relevant treatment options are so you can plan for when the patient recurs. You don't have to wait to do additional testing and wait 4 weeks to decide what next phase is, but also to predetermine eligibility for clinical trials. One of the biggest reasons patients don't enroll in clinical trials, well, first of all, they're not identified. About 60% of clinical trials today require a biomarker. But even if they have testing done, if they're not pre-qualified, they don't want to wait another month to do all the testing to see if they're eligible. And so they go on to another treatment. And that has a lot of consequences. It deprives patients of contributing to science, contributing to the advancement of scientific knowledge, and contributing to enrollment and accrual of patients in clinical trials to help establish whether or not this new therapy has benefit for society as a whole.
And I guess it begs the question, is that the payer's role to cover that larger panel so that the patient can enroll in a clinical trial that, again, the payer may not see any benefit from that? Downstream costs may still end up being high if the patient doesn't respond to therapy or if the drug doesn't end up working or whatever the case is, that payers are going to question the appropriateness of those larger panels just to determine if the patient should be enrolled in a clinical study.
I've had oncologists say to me, well, if I think a patient's eligible for a trial, well, I'll test them for that biomarker. But in community practice, that's not the question. The question is, what trial is the patient eligible for? Some payers will deny that as experimental investigational. There's nothing experimental or investigational about identifying a biomarker. We have analytic validation techniques. They may say that it doesn't have clinical utility, but that gets back to our discussion earlier, is identifying all the relevant treatment options for a patient. Does that have clinical utility? I would argue yes. Payers may say no, but just on the economic side of things, payers think that clinical trials cost more money. The published evidence suggests that that's not the case. And certainly even in Medicare Advantage populations, all trial costs, the routine trial costs, revert to original Medicare. So when I was practicing a health plan, we tried to get every patient who was interested to participate in a cancer clinical trial because all those costs reverted to original Medicare.
Oh, that's very interesting. So it's not so much just about the immediate cost of the test, which is admittedly relatively nominal compared to downstream targeted treatment costs, right?
Yeah.
It's also about whether or not that is going to make a difference for the payer in the long run. It's one of those things where we have to consider the benefit to the patient, not just the financial cost of the treatment and the long-term outcomes that we're seeking with when covering these tests. So again, I think this is an overlap between the understanding of the economic feasibility of these tests and also the clinical utility. So I wanted to bring it back to clinical utility as we start to wrap up here. And that is with a couple questions that we try to do on a regular basis here on the Unscripted podcast. We want to know what your unscripted truth is about clinical utility. What do payers need to know that they don't know right now?
Well, with respect to this topic and cancer, this limitation of access to testing can have a profound impact. And it's not all payers too. There are physicians who don't order testing, and then there are insurers that limit access to testing, but there's a consequence to that. There are 5 studies published in non-small cell lung cancer that have shown that patients who are appropriate for targeted therapy but don't get it die prematurely, and they die somewhere between 8 and 14 months prematurely. And if you look at the estimate of new cancer cases, about 92,000 patients with metastatic disease. If you look at the proportion of patients who don't get testing, about 20,000 of those will die prematurely every year because they either didn't get testing or they had testing done, but they didn't get matched treatment. There's a consequence to our failure to deliver evidence-based care. And the consequence is not only premature death, but also wastage from the wrong therapy.
That's a really good point. I feel like we should emphasize that the payers are not necessarily not covering biomarker testing that of some sort of nefarious approach to save money, right? I think what they just want to know is whether those downstream outcomes are going to be improved through this test. Because like we talked about, the majority of the tests that we're seeing for oncology in particular are lab developed tests. And again, I don't think there's any question that payers are looking at the FDA approved companion diagnostics and covering those without question. I think it's more along the lines of there are lab developed tests that can test for a lot of different things, and we just don't know what the clinical value of testing for that is exactly, right? In terms of improved outcomes. It comes back to this idea, like you said, if we are covering evidence-based medicine, now the evidence-based tests, evidence-based therapies, then we're doing our job. And I don't think it's inappropriate whatsoever to make sure that the evidence is there, right?
I wholeheartedly agree. And in my role as a health plan medical director, I didn't cover something unless that there was evidence of that clinical utility.
Yeah, exactly. And so then it becomes the question of how do we define clinical utility? How do we agree upon those outcomes between the different stakeholders, not just the biotech companies and the payer, but also the patients, the caregivers, the providers, even pathologists, et cetera. So I think this is a really important take-home message. And as we leave with one final question here, we try to leave on a positive note. What's one silver lining that you can take away from stakeholders having different perspectives of clinical utility?
Well, I'm going to give you 2. We don't know today how many patients are benefiting or not benefiting from precision medicine because we don't have a method for assessing who's been tested and who hasn't. The U.S. Oncology Network developed a measure several years ago on non-small cell lung cancer to assess which patients had adequate testing and which patients had test concordant treatment results. ASCO is working on a national quality metric now that we hope will be adopted by Medicare so that we can answer at any point in time, how are we doing, but also, are we making progress? I think the second thing that I'd like to share, and this is specifically for AMCP members, is that there is something that each health plan and each health plan pharmacist can do to ensure that patients are receiving optimal care. And that's during the prior authorization process. For example, when you're getting a request for pembrolizumab for a treatment of a cancer, ask the question, has the patient had adequate or full molecular diagnostic testing to determine that that's the drug they're most likely to respond to? I know that Evacor, which is a laboratory benefits management company, did a study several years ago that showed that they could actually reduce the amount of inappropriate therapy being delivered and increase the appropriate testing being done by just asking that question. And I think that's something that every practicing pharmacist at a health plan can implement within their health plan to ensure that patients get optimal treatment, aren't harmed by the wrong treatment, and aren't spending money on the wrong treatment.
Yeah, I think that's a very simple take-home question. Just considering, is this the right drug for the patient, considering all of the testing that is potentially appropriate for that cancer type? And what that patient has undergone so far and all of that. I think there is a lot more that we can discuss, but unfortunately, I think we are out of time. So Dr. Fox, thank you so much for your time today and all of your insights. And thank you to our audience for listening.
Thank you very much, Steve. I appreciate your time, and I appreciate the opportunity to contribute to the dialogue on this topic.